We got this data, we got the fast track. Investigators like to use the drug so far. What we want to do in the next study, it's a global study. We're going to pursue it in the US, Europe, and Latin America, around 125 patients.
Your market cap is around $250 million.
We grew our shareholder base with 25% last year, so we're around 14,000 shareholders.
I'm always curious because I think over the last 10 years, first, I haven't heard about too many success stories in repurposing, but I've heard quite a few failures as well, and I've heard quite a few pushbacks, especially from VCs who don't like it too much, pharmas don't like it too much.
We defined the opportunity that we could improve survival with this old drug if it was used differently in heart failure. What happened with this study was that we improved survival with more than 30%. Published New England Journal of Medicine, great success.
Bienvenue to a new episode. I'm your host, Philip, and on the Flot.bio show, I'm interviewing the best Europeans in biotech to help you grow. Getting a positive phase 2A results in a high unmet medical need as an independent biotech is quite an achievement. Cereno Scientific recently reached it. So I caught up with its CEO, Sten, while I was in Lund in the southwest of Sweden. I had never met Sten, but I followed him and Cereno for the past 2 years upcoming to the results. We talked about the next steps of Cereno, including the upcoming phase 2B. We also talked about how the company reached one of the largest retail investor communities in Sweden, and also talked about success stories in drug repurposing. For transparency, this episode has been sponsored by Cereno. So here's my conversation with Sten, and please hit the like, follow button if you're enjoying it. Good. Welcome to the show, Sten.
Thank you, Philip. Good to be here.
Yeah, it's good. It's good.
Yeah.
So I want to start with Cereno. I mean, you had really nice phase 2A results. You are starting your phase 2B, the first enrollment very soon. We'll talk about this. But actually, I want to start, and I haven't found too much about the origin of the molecule and where the molecule come from. So maybe you can start with that. Just tell me more about it.
Yeah, sure. Again, we're super excited about the stage we're in as a company, and we really believe we have something here. But it started a long time ago for the molecule. So sometime late 1800s in Swedish, 1880, 90, the molecule was discovered as a molecule. It was later applied and eventually launched for neurological diseases. And the major use has been for prevention of epilepsy. And that's been the case until fairly late. It's been applied also for cancer, together with other therapies. But never really pursued into what we are doing. We're very excited about the development of knowledge around that type of molecule. When we did our review, there are 600 articles about HDAC inhibition, most of them VPA, which is our molecule. And a lot of documentation on other things than neurological disorders that are very exciting as a platform. And we believe that applying HDAC inhibition, which this molecule is able to do,
histone
deacetylase inhibition,
epigenetic modulation.
Exactly. So about 30 years ago, if I'm fairly correct, that capacity that the molecule was an HDAC inhibitor was discovered. What does that mean? Well, it can inhibit histone deacetylase. And what is that? Well, DNA, as we know, is the map of the protein production in the body. But it's a map for the factory of protein production. But around this DNA are regulating enzymes, and HDAC are one of those abilities to regulate the production. And that's all fine when it works well. But if it doesn't work well, something goes wrong in the production, and that can have serious impact on the body's ability to function well. And we can maybe address that a little later in the talk.
Okay. And then I'm curious if the molecule was discovered so late, so how do you build IP around it?
What you can do, of course, when you discover that, this molecule has a different capacity than what has been applied for and patented for and used for a long time, you can, of course, apply for a use patent around the molecule, which we have done at Cereno. We have also developed and produced a very different administration form, a packaging and administration form that fits the needs that we believe we have in order to get the efficacy we want. So we have 3 patent families around the drug that we have applied in all the markets which we pursue, and most of the key markets we already have patent. In addition, if you want to protect a molecule like this for a new use, if you are pursuing an orphan disease, a rare disease, you could, if you have enough data and the authorities, FDA, believe that you have something, you could apply for orphan disease designation. If you get that accepted designation, you will have in the US 7 years protection, exclusivity, and in Europe 10 years. So we have done that. We've done that also. So we have it both in Europe and the US for this molecule, which we call CS1, which has VPA inside it.
Okay, that makes sense. Yeah, and so the main indication is pulmonary fibrosis, or PAH, and that's where you got the phase 2A data. Can you talk a bit more about the data and the results?
Sure. So what we did when we found all this documentation with numerous proof of concepts in various animal models, etc., we decided that we should pick the disease that we believed had the highest impact from our point of view, with our capacity and the mode of action that we have, with the least time and least capital effort to get the work done and be able to help patients. So we picked the orphan disease pulmonary arterial hypertension, which is actually not mainly a fibrotic disease. It is most affecting females, and upon diagnosis, they have about 7 years to live. And what happens is that they die of right heart failure. And why is that? So the diagnosis is really that these females have a hard time getting oxygen to their lungs. And the reason is that as they live, the pulmonary artery between the right heart and the lungs, the connection, the tube, the vessel, where the heart is supposed to pump blood and oxygenate the blood and then flow back to the right side and pump out into the body. That pulmonary artery, the myocyte inside, the muscle inside, grow to produce a more narrow tube. And there's also a fibrotic element, connective tissue. So this tube that becomes more narrow also becomes more less elastic. So it becomes more narrow and narrow tube over time, less elastic, and that provides a very hard resistance for the right heart to pump through the blood. So the right heart hypertrophies and eventually collapses of too much work. So you go from an early diagnosis when you maybe are a little tired when you do exercise. That's New York Heart Classification 1 of heart failure. And through class 2, class 3, and class 4, where you're bedridden, you can't really move. And through these phases, the heart becomes less functionable. So what do you do with these patients? You try, up to now, you've helped these patients by dilating the vessel, the pulmonary artery, and that's a temporary relief. It has helped them to go from around 2.5 years to 7 years life expectancy from diagnosis. But at the end of the day, you don't reduce the progression of the muscle growth and the connective tissue growth. So at the end, this heart still collapses and you die after 7 years. So what everybody is looking for is something that can slow down, halt, and even reverse this progression for these patients.
More from a systemic point of view rather than a like a physical.
Rather than a physical hemodynamic point of view, of course you want the hemodynamic
both, I guess.
Yeah, you want both, and of course you'd like to do that very quickly. But in the long run, you need a disease-modifying agent that can impact this progression, halt it, and maybe reverse it. And that's what we believe that we have. This is what has been documented in animal proof of concept studies. They're done in rats, where you can see that you can prevent the progression to PH by introducing the agent early. If you introduce it when you have established PH in these animal models, you can reverse it. So that's very encouraging, and that's one of the reasons that we picked this disease, that data was already there. So we have then taken this, our HDAC inhibitor, and pursued this indication for these patients. And we got some very encouraging data out of our trial that we pursued after FDA acceptance, phase 2 trial. We pursued it in 10 centers in the US in 25 patients. And we presented the data last year, in the beginning of last year. And it was very encouraging for us. And I could maybe mention some of the data that we got in our trial.
Definitely. I mean, I don't even have the date, but
So
it was a phase 2A, and you had favorable safety and tolerability, and basically positive results.
So this was a fairly small trial. We added the drug on top of standard of care. And they should be stable in their disease over a 3 months period. And they should be in New York Heart classification 2 and 3, so fairly sick patients. And we added our therapy and we followed them for 3 months. And what we saw after these 3 months was that we had an impact in 44% of the patients, and the evaluable patients were 21, so it's a small group. But in 9 of 21 patients, we improved the prognostic value. Reveal risk score is a tool you use as a doctor to value the prognosis of the patient and the risk of dying, actually. So we improved that more than 1 point in this scale that is used, which means that over a year, we improved the risk of mortality with 24%. So they lessened the risk of mortality. And then we also looked at New York Heart classification, this class that they were in. And after only 3 months on top of standard of care, we moved 1/3 of the patients to a less severe class. So 5 patients were moved from class 3 heart failure to 2, and 2 patients from class 2 to 1. You almost not feel the disease.
You really reversed.
We reversed their functional class, so how their heart was functioning for them. And in addition, 2/3 of the patient, we reduced the pulmonary pressure on. We worked with Abbott with their implantable device, so wirelessly we could measure the pressure with this device. And in 2/3, we reduced the pressure, which is also a good signal. What was maybe even most intriguing here was that after this 3 months, we saw an improvement of the right heart to contract. That is the strongest risk prognostic indicator for these patients. How well can the right heart contract, pump the blood into the lungs? So we had a significant improvement of this global longitudinal strain factor, which is what is called. Then, of course, FDA released an article, I think it was in March or April last year, about what's expected of drugs that want to help PH patients now. What do we expect? What do you want them to do? Well, they want to have a disease-modifying agent, and they want to see impact on prognosis, classification of heart failure, hospitalizations, and they want to see that these patients actually feel better. Because that also is a very strong goal from the regulatory authorities when you introduce new drugs. And we did both heart failure, the Missouri questionnaire, and specific for pH Sympact. And we had an improvement between 30% to 85% in these questionnaires of the well-being that they expressed after taking our drug. So all in all, very exciting data for us. We think we're seeing the effect of what was documented in the animals now in patients for the first time. So we are now embarking on
the phase 2B.
Phase 2B.
You just received, in December you received, what was it, approval from or clearance from the FDA?
Yes.
And then you will start.
Exactly. So, but before then, actually, even, what happened during this study was that the investigators asked us, when they were done studying the patients, all the data was in, they said, we don't want to stop taking your therapy or giving your therapy to our patients. So please submit a compassionate use application to the FDA so we can continue. So we did that, and we got that accepted by the FDA. So now a number of patients, 10, were enrolled in this compassionate use program. So they got to use our drug for a year longer. And we haven't had readout on that data yet, but we have supplied these investigators the drug, and the patients have had the drug for a year beyond the trial, which is interesting specifically for safety and tolerance reasons, I would say. We might also see some efficacy data, but specifically safety and tolerance is important for this drug in this patient group. And then, of course, after this data was presented and we had a meeting with FDA, Type C meeting, and declared our ambitions and the data. We got the feedback from them. And then we also actually got fast track designation by FDA in September last year.
So you have orphan drug designation and fast track?
Yes, exactly. So we feel we have the nod from FDA, we think you have something here. That's why you get fast track. We will work with you in a good manner to get this as well and as fast as possible to the patients. Of course, providing that you continue to show the efficacy and then document safety and good tolerance on the drug.
And then I saw, first patient in, you're expecting, I think, Q2 2026, and then top line data Q4, end of 2028, and 126 patients. I mean, I'm reading the figure that was all in the press release, but just to have, the exact number, but to have an idea. That's correct, right?
Yeah, that's correct. And I think, so we got this data, we got the fast track. The investigators like to use the drug so far. We have compassionate use going on. It's actually stopped, and we're going to analyze the data. And what we want to do in the next study, it is a study. It's going to be around 60 sites. It's a global study. We're going to pursue it in the US, Europe, and Latin America. Around 125 patients. And the study design is very much in line with what is now the modern way of documenting in PH, which is a longer study. So we're going to do 36 weeks. And we're going to have 2 dosages and 1 placebo arm, 36 weeks. Then we're going to do distribution of, so those who have placebo will get one of the 2 dosages that are active. And then we're going to continue for another 24 weeks. So it's a longer study. It's a year. It's a longer study, and that will provide us the ability to document the disease-modifying capacity that we want to capture. And it also provides for all the patients to get active drug. So not one group and that are going to be only on inactive treatment. You also can have a greater ability to potentially see if the data that you have will linger on, which would be a signal of disease-modifying capacity also.
And why do you need 60 plus centers if you want 120 patients?
Yeah, it's a good question. It's a rare disease.
Okay.
So that's one reason. That's the main reason. Yeah. And we want to be fast when it comes to the recruitment. We have learned our lesson. And then the other point is that there are other trials going on at the same time in the world in this patient group. So you want to have a broad enough fishing net to bring in capture patients that would like to be in the trial. And that's why it's designed that way.
Okay. And I think I heard that you were maybe hoping that there would be a phase 2b/3, but not doing phase 2b. That's correct. So it's
not really.
Okay.
Well, we had a strategic thinking around what should we do? What's smartest to do here? Should we pursue 2b/3 trial or a 2b trial? So we hadn't decided yet, but we said that's a possibility. And then when we took a hard look at that after the data and then realizing that if you do a phase 2b/3 trial, you're stuck with a certain design upfront. And so the flexibility of a phase 2b trial is a better setup to run this program on. It provides, we believe, a stronger possibility of positive outcome of the data than if you do a phase 2b/3, because you move yourself into locked situation for a both phase 2b and a 3 outcome. You don't do that if you do phase 2b first. So that's why. And we also, one reason is, of course, strategic for us. We believe that we think that the study will be positive, both from safety and tolerance. We think we will have the best drug in terms of safety or tolerance when we come out. The drugs out there are quite nasty to take, including the latest one, sotatercept or Winrevair. It has its drawbacks and also safety risks. So this will have been in man forever, 60 years plus. In high dosages, we're using low dosages, an oral drug. So safety and tolerance and administration will be very good. We believe that if we can document the efficacy that we're after, that we will do a deal either before the trial is done or when the phase 2b trial comes out, because that will be a very attractive proposition, we believe. So we don't want to. So the journey of a biotech, as you know, is either you go all the way to market or you go to do a deal. We believe this setup is the best setup to get to a deal. So that's another strategic reason. Yeah.
Makes sense. I mean, it's what you just said, but it's very rare, especially for European biotech, to bring their first assets to commercial.
Yes, it is.
I don't know.
No.
So many Argenx, maybe the big exception.
Yeah, Calliditas actually did it.
Calliditas.
Yeah, and that was a repurposing. So that was budesonide in a different disease.
René was on the show.
They were later acquired.
René was on the show. So she had
René. Yeah, I know her well. Yeah.
I mean, now she joined Hansa, but and she was already CEO of Hansa. Yeah. But Argenx, I think, is even different scale.
It's a different scale. Actually, we have, a mentor to me and later to Cereno the last years is Don de Behitzi. He's vice chairman of Argenx. Okay. So I have had a good relationship with him. He lives in Copenhagen.
Okay.
So we meet now and then, and he's actually an advisor both to the board and to operations nowadays.
Okay.
So he has a good...
big person.
Yeah. He has a good empiric knowledge. So
can imagine.
Yeah.
Okay, that was a little side chat.
Yeah, sure.
But what you mentioned competition, I had it listed because I think Merck or American Merck the drug is approved, was a first in class, disease modifying as well. I mean, you said you have some side effects, but I guess from a market capture, I guess they have a pretty good position.
Yeah, absolutely. So, one thing that struck us when we looked at the data around VPA and then, we call it CS1 now because it's repurposed, but was that we found sotatercept preclinical work and we found the work on VPA. And they were very similar.
Okay.
So we said, hey, Merck just bought Acceleron for $11.5 billion. And
that's the Winrevair drug of the
sotatercept was in that deal valued to $7 billion. And sotatercept had just done their phase 2B program, finalized. So they bought that asset at that time based on the work that had been done in preclinical and then in the phase 2 program. And we said, well, wait a second. We have preclinical data that is very different. We have a very different mode of action. And, then Merck pursued the rest of the program. And they've had great success with their sales. And I think projected sales are like $7 billion per year now, exceeding expectations. The reason is there's such...
Where is it today or 2024? What the sales?
No, I think it projected peak sales.
Yeah, but where is it at today?
I don't have the latest figures, but they are exceeding analyst expectations so far. The reason, of course, is the huge need for a different kind of therapy than vasodilation.
Yes. Yeah.
Realizing that sotatercept is a TGF-beta monoclonal trap molecule. So what is that? Well, TGF-beta is a promoter of changes in relation to pH. It's an inflammatory promoter. Now, monoclonal antibodies are not necessarily often that nice to take as a patient. And what happened to another company, Keros Therapeutics, that had a drug that is a bit similar to sotatercept, they crashed the phase 2B program because they had pericardiac effusion, so fluid in the heart, in the heart wheel. So, and they stopped the whole development. Now, sotatercept as the monoclonal TGF-beta trap molecule has currently to be taken as an injection. It's
IV.
Yes.
In the hospital?
A syringe.
Okay, syringe, yeah.
So injection every third week. And what has been documented on the side effect side and the safety side is there has been fatal bleedings. There's a risk for either bleed or thrombosis. You have had pericardiac infusion incidents, and there are other side effects or tolerance issues. So this drug has this combination of being quite effective for these patients, up at least to a 6 months period, but has its risks. So we know that if we succeed with our efficacy in our trial, that we will be a very good addition to the therapeutic arsenal that the physicians have, and that it can be used, we believe, together with Winrevair. It can be used with the others. And this is very similar to what happened in heart failure over the last 20, 30 years. That originally patients in heart failure was a pump and plumbing exercise, so with drugs. So diuretics to get fluid out, inotropic agents to help the heart pump, and so on. And then there was a development of hormonal control that improved survival and well-being for these patients and extended their period of living. Pulmonary arterial hypertension ends in right heart failure. That's how you die. This development of new drugs for PH is 30 years behind heart failure development of drugs, we believe. So, and the people involved with Cereno were involved in development of heart failure drugs, and I've been part of that journey. So we believe now that by adding new agents to PH, we will be able to extend the life and well-being of these patients, and that the oral administration of an HDAC inhibitor, such as us, will do that job. Okay. And what we believe we will be able to do is actually slow down, maybe halt the progression of this muscular fibrotic development that is narrowing the pulmonary artery.
And in terms of efficacy, you're expecting similar level of efficacy with Winrevair, even if you're oral versus a MAB, like an injected MAB?
Yeah, I think we will have, it's difficult to gauge the level of efficacy, but one will, when you look at the drug and its possibility for approval, so far FDA is looking at the additional efficacy that you can give on standard of care.
Yes.
So we believe we'll add additional efficacy to the standard of care, which will be then vasodilators, and in some countries, maybe Winrevair or sotatercept. It's not yet. So but it will be part of our trial.
Okay.
And we believe we'll have the efficacy that is required from regulatory authorities to approve the drug together with other agents.
Okay.
It's similar to what sotatercept was doing. So they got it approved on top of other agents. Yeah.
Okay. I like what you mentioned with the team and how you were involved. And before we hit record, you told me also about the repurposing. I want to talk about that a bit later. But first, I want to finish a bit on the pipeline as well, because you have a second product. So I think it's CS014 or 14.
Yeah, CSO14 we call it.
CSO14.
Yeah.
And you had the positive phase 1 result, I think mid last year, right?
Yeah. It's just, why are we doing that one?
Yeah.
So, because it's also an HDAC inhibitor.
Yeah.
And when we realized all of the proof of concept data that's there on HDAC inhibitor, especially on VPA, in pulmonary and cardiovascular diseases, and that hasn't been pursued into man and as valuable drugs to help these patients, either with well-being or prognosis, progression, we decided quite early that, okay, we'll take VPA, we repackage it to an administration that is a good one, and we protect it with our patent families. We have 3, and we protect with orphan disease designation for the first indication, that's PH. But we said this potential of this concept is too big for this first attempt. We want to produce a new chemical entity that's also optimized from the first one, still capturing the efficacy capacity of the first molecule. Okay. So we decided to pursue a second generation, and that's CSO14. Okay. And it's actually the same molecule, but we have optimized it by deuterizing it in 2 positions of the molecule. And why did we do that? Well, we wanted to get rid of a metabolite that BPA has. Yeah. And we have basically abolished that metabolite route. And why that metabolite? Well, if applied to children, it's toxic for the liver. So 1 in 20,000 kids get liver toxicity. That's not a problem for PH, of course. But if you want to pursue many different indications, why not do a new molecule? And why not do one that has the new chemical entity protection? And also, some pharma companies prefer new chemical entities, so that will be a broader and for some, a more attractive proposition. Okay. Now, CS014 is pursued for a different purpose than CS1. The purpose is lung fibrosis.
Yeah, IPF, right?
Yes, yeah. IPS. So why can we do that when we're pursuing muscular disease progression, pathological one, mainly in the pulmonary arterial hypertension? Well, because there are also proof of concept studies on the lung fibrosis models with HDAC inhibition.
Okay.
So we now decided to go for another orphan disease, which is IPF, idiopathic pulmonary fibrosis, with our second drug. That way, we are documenting the ability of disease modification in 2 different orphan diseases that has already proof of concept in animal work.
Okay.
And that gives us, of course, 2 attempts to succeed. And it's hedging the attempt, but it's also 2 very unmet medical need markets.
Okay.
Now, IPF is affecting mostly men compared to PH, which is mostly females. So that's why we're doing CS014, what it is, and why we're choosing IPF.
Okay. And in IPF, actually, I had on the show the CEO of In Silico, and I don't know if you know him.
I don't know him.
The company, but they have, I think, phase 2A results in IPF as well.
Yeah. Good. But there are almost no drugs. There are a couple of drugs. They have very bad safety and tolerance problems, and they haven't really proven either symptomatic or prognostic relief. Okay. So, but that's what's there. So the market needs new things, and we hope we can help. Any company that's succeeding in these, we applaud them. They need it.
Okay, that's cool. And I think, if people want to watch, so it's In Silico, and what's actually quite cool about them and quite unique is that it's the first AI generated from scratch that has gotten efficacy data. It's the first globally.
Oh, it is. Cool.
Was actually
I've read about it, but I don't have it there actually in my head.
Alex Savonkorov, I think is that name.
Where's the company based?
So headquartered in Boston, but quite global.
Yeah.
With quite a base in China as well.
Yeah.
And they just IPO'd on Hong Kong Stock Exchange. I think 2 or 3 weeks ago they raised $300 million.
Good.
And check it out.
They're paid to do it.
Good.
And so I should say also that I think that CS014 might we're actually looking at it because. What we did with CS014, we just published the original idea with HDAC inhibition, epigenetic modulation, that was the idea of the founder, Professor Sverker Järn and some of his colleagues in 2012, was that you could actually prevent thrombosis in man without increasing bleeding risk. And that has been documented with CS014. We just published it. So that is still in long-term possibility with this approach. It would be fantastic. Thrombosis is the biggest killer on the planet. All causes that are non-communicable, so non-infection and things like that. So So that is still a possibility with this concept. But if we look at our work that we did with CS014, we actually repeated a pH study with CS014. There is a study or two studies with VPA. And what we saw with CS014 was that we had impact on plexiform lesions. And that's actually the very root cause of the small arteries in the lung, that we had an impact on those dose-dependent impact on those and on the fibrotic development. So it's not yet published. We have talked about it, but not yet published. So that strengthens our belief that there's a possibility for disease modification. And we have actually considered to pursue CS014 in the adjacent disease, PH-ILD, interstitial lung disease, so pulmonary hypertension, interstitial lung disease. There is a fraction of those that have IPF, but PH-ILD is something that, we might pursue as we move forward. So there are several avenues here.
Yeah. Some potential for more. I'm curious on also doing two later stage trials from a finance point of view. I mean, it's expensive to run a phase 2b.
It is, yes. I agree. Yeah. So, we are...
But you're quite well financed. So the fundraising you made, I think your market cap is around $250 million as we talk around there.
Yeah. So we grew our shareholder base with 25%, around 25% last year. So we have around 14,000 shareholders. Yeah. We have fairly good liquidity in the stock for our size and being in Sweden. We are NASDAQ First North. And then we grew our market cap last year with around close to 30%, 27%, I think it was. We were actually beyond 3 billion Swedish. And over the fall, it's gone down again to 2 billion. Still a sizable company in the market. So we have, in a way, gone against the stream after 21, when most biotechs had a reduction in 70 or more % in the market cap. So I think we have a strong following and a growing following on the stock market. We've been able to finance the company in a good way, I think, so far. It's been a tough market for the last 5 years or 4 years. So we've had a growth period then. I think it's because we've been able to deliver on what we have said and maybe a little beyond that. And that there's a very visible bench in the WinRiver growth and deal that Merck did with Acceleron and valuation there. So if we succeed, I think the investors believe that they can get, a good return on their investment, and that's why they're with us and increasing. That doesn't take away that it's a challenging financing market. So we recently did debt equity financing, which we did at a premium, a slight premium. And so that by itself requires the share price to go to a certain level for the conversion of the debt financing we have and the next one to come in, and also for warrant packages to come in. So we are, carefully looking at our development, and we will address if we need to do more to get to the finish line. So that's where we are.
Okay. But so you think you will be financed to at least finish the phase 2b to get the top line data?
If the current program...
And not with the current cash, but you have...
No, with the current structure that we have. Yeah. And if for some reason that won't work out, we'll address that and do more financing.
Okay.
So...
How much cash you have on the balance sheet? The latest data?
Yeah, that's a difficult one because the Q4 is not released yet. That's on the 28th. Yeah, we raised 100 million in equity, and we refinanced the loan. So I think
100 million SEK, so
it's Swedish, so around 12 dollars.
Yeah.
Dollars gone down.
yeah.
We have a lot of expenses in dollars. That's a good thing for us. But yeah, so I don't think I'm well read enough to qualify on what.
I mean, I think we can edit and add at least the Q3 figure.
Yeah.
And, okay. But I guess enough to move
forward.
Move forward and finance. Okay. And talking still about finance investors, I'm curious on your investor base. I mean, you said there's 14,000. I think you have a quite strong also retail investor base.
Yes,
in Swedish.
Yeah, we have. I mean, in term of when I joined the company on the board 2014, and it was an idea, really no money, one animal study going, and no real business plan. And I've been in biotechs and pharma before, so I knew what was needed, and we needed cash. We needed a good business plan and cash for this fantastic opportunity, which we can come back to later why I thought so already then. I've been 11 years on this now, so but the investor base, the choices at that time was, no patent, no money, no real data more than the very early data, experimental data, and some animal work, and no VCs really present. HealthCap was here. The others gone after the last recession, which was...
HealthCap invested.
No, HealthCap didn't invest. Okay. No, we didn't even go to them at that time. But so we decided to, let's go to the stock market. It was open. People were interested in going in. So we put together a prospectus, and, after 9 months, we had 30 million Swedish in the bank and a valuation of 84 million Swedish from zero. So that was a good start for us. And we are on that road, I would say. So what has then happened over time is that the investor base around 4 years ago,
so you IPO'd kind of at the $8 million valuation, and now you're at the $250 million valuation.
Yes. Oh. But again, this was
it took some time, but
2016.
Yeah.
But I think that what happened about 4.5 years ago, we got some investors interested in the company, and they started to really read up on our information, including what I mentioned before here, all the preclinical data that's available and our data. And then, said, we want to stay here long term. And this group has actually created something called a Discord. So a space where they store all the data on the company. And it's a fantastic place. They have a lot of maps on patents, science, publications, everything that's going on with the company. A very good competitive intelligence source for me, because there are people there working day and night to source the information and comment on it, etc. And I think this was the start of people knowing the company, understanding it, understanding science. And there are cardiologists, people working with heart failure patients, etc. They're there. There are investors, etc. So I think that was the start of that. And that group is now close to 5,000 people.
Wow.
And there are a group of investors there that are very strong and has been with us for a long time. And I think we had 12 investors in our last equity raise together with the debt financing, and they're part of that group.
Okay.
Very convinced of our potential and our future success.
Yeah, because I saw actually I have two things I saw on this. One is actually I think 2 or 3 of retail investors actually messaged me
Yeah, they're very active. They want to. Are you going to do an interview?
Yeah, you need to have Sten on show. Like, no other. Like, I haven't received a message like this
from
any other.
They're very, very interested.
Crazy.
Yeah, they're very interested, and I like the interest. It's a double-edged sword because sometimes I can't answer mail from 13,000 people. I would do nothing else. I wouldn't even do that well, I think. So it's a double-edged sword to have such a following, I think. But in general, it's a very good thing. Yeah. It's similar to you. You have 30,000 followers, right? You want followers if you are on a quest to change the world and to change the paradigm in these orphan diseases. So I like it a lot. But back to our investor base, that's our investor base. Yeah. And of course, as we move forward, we would like to attract other types of investors.
Institutional, I guess.
Even more high net worth, family office, or other types of investors, such as potential pharma partners, either local, regional, or so, or global, or more of the VC-oriented that like to go in when there is data enough that they believe this is going to fly. So that's the future. Yeah.
Sounds good. I want to finish more on the team, and then it will be a good transition, I think, also to your own experience, because I think you have like pretty strong team. Yourself, and also I saw that your CMO was actually a CMO of cardio. Claudia, the CEO, was on the show, and cardio was a really nice exit to Novo Nordisk.
Exactly.
I think a bit more than 1 billion.
Yeah, 1 billion euros. So
euros. I don't know, and I didn't manage to get the upfront, but I heard it was very sizable upfront.
Thomas Munn. He's
I don't know, founder, CSO, I guess.
He was founder. He's the founder of Uni.
And then I think your CSO was there also before you joined at Cereno.
Yeah, it was at Cereno. Was on the board. Was Jonas Frerson-Selje and Björn Dahlöf. Jonas and Björn joined the founders first, already in 2012 when the company was founded together with Göteborgs University Ventures.
Okay.
And they were the first investor with a number of 100,000.
That's why the company is based in Gothenburg as well, I guess.
Yeah, and Gothenburg is the foundation of AstraZeneca's success in cardiovascular disease and then GI, and their collaboration with universities and hospitals.
It was the headquarter of Astra's research, right?
Well, I worked for Astra globally. So, and also my first job was a sales rep for Astra. But Astra had actually 5 research units once. But originally Astra was in Lund for respiratory diseases, in Gothenburg for cardiovascular and then GI, and in Södertälje, close to Stockholm, for pain and CNS.
Yes. Okay.
And they were very, the philosophy was like most pharma today, was to establish and work very closely with the university. And so that then sprang out to success in various disease areas. But then again, of course, Sverker Järn, Professor Sverker Järn and his colleagues were based at Sahlgrenska in Gothenburg. So that's the reason they founded the company there. But it's a good position to be for this kind of company. We moved into Astra's BioVenture Hub when we were very young, when we listed the company. And then we have moved out to more grown-up research facility platform, which is called GoCo, which is next to AstraZeneca's facilities, just when you drive into Gothenburg in the village Mölndal, just before that. And we have a very nice facility there. And I think there are around 60 companies there. Of sizable, including Repurposing, that just listed on Hong Kong Stock Exchange.
Yeah.
So so
I was about to ask you a bit about Gothenburg, but we can talk about Gothenburg. But
so Björn is currently the chief scientific officer and has been for a long time now, since around 2018, I think. And then we have Rahul Agrawal, Dr. Rahul Agrawal. Björn is a cardiologist and used to be a professor in Gothenburg. And Rahul is actually, he started as a cardiologist, joined Menarini, and then was recruited over to Bayer, ran there, among other things, the rejuvenation of pH on the market. And then he was recruited into top-level positions at AstraZeneca and reporting directly to Pascal Soriot for his omega-3 venture. So he has had, he's had 250 patients in clinical trials worldwide. So Rahul is, when I got the opportunity to get my hands on Rahul, so to speak, I was very happy. He's told me that I'm the only one that took reference on him in his career. So, but that's a side story. But he was with Cardior. He left Astra for Cardior, and then Cardior was bought by Novo Nordisk. And just in that time, I got contact with him, and we were happy to join forces. So he's head of R&D and CMO for Cereno.
Yeah, that's cool.
It's a very good team, and
that's solid. I think we've done quite a good tour of Cereno, and maybe switching more to industry topics. I mean, we talked about Gothenburg and the Swedish biotech ecosystem. I want to maybe expand a bit there. Gothenburg, I didn't know too many companies. I know quite a few in Lund, quite a few in Stockholm. And also what I just realized coming to Lund was that the universities, I knew Chalmers, of course I know Karolinska, but that Lund is actually better than Chalmers, or like very good also in kind of top 3, and that they are quite, I don't know, equilibrated, I don't know if it's a term, but they are ranking quite closely. At least Lund and Karolinska, I think they're quite close, and Chalmers is also a very good university, versus, I don't know, in some other countries where it's like number 1 is clearly ahead.
There's one, yeah. No, here's a good spread, I think. And I think, then one could also, of course, look at, what are good platforms for growth. I think one of them has been for Sweden, and is the ability for an inventor to own the patent.
Yes.
So do research such as in our case with Cereno, and be able to patent your research. It's not captured by the hospital, which is the case in the US. And we haven't spent
almost everywhere in the world.
Everywhere. So there is an entrepreneurial spirit. That's also a double-edged sword, because then you have to be able to capture people with experience on the venture. It's not that easy for a professor to drive a company to success. So that person needs to be surrounded by knowledge and capacity to get it done. And the journey is...
Frederick Cameras managed from professor to CEO, but it's quite rare.
It's quite rare, and there are some people that are brilliant on everything they do. So all respect for that. But I think, so if one wants to look at the Nordic area, maybe Sweden, one thing that happened here was that AstraZeneca actually closed down the research unit here in Lund. And so that opened up for a facility that could be occupied by entrepreneurs. And also Lund was very early with the incubators for other types of startups. Ideon, which is next to Medicum Village, which is the old Astra respiratory research facility and marketing facility. So by good intentions and good strategic thinking, Lund and an entrepreneur from a real estate company joined forces here and built Medicum Village infrastructure and ambition for the entrepreneurs. And I think fairly short after, the whole facility was filled, and has had to expand. So I think that is similarly happening in Gothenburg. Yeah. You've had these holding companies, Chalmers Ventures and Göteborg University Ventures, being incubator promoters for startups, helping the startup people, including professors, etc., in their fields, and then getting them on their way. And the trick is then to have people such as maybe myself that has been in big pharma and in other startups to join the venture and to get it going. So I think that is present at around Karolinska Institute, Chalmers and Göteborgs University, and down here in Lund. Also, there's a growing hub in Uppsala.
Yeah. But Uppsala is very close to Stockholm.
Yeah, so it's, everybody wants their own identity.
I guess it's like Malmö and Lund.
Yeah, a bit.
But,
Lund has always been way back the center of knowledge based on the university. Later on, Malmö Högskola, was established and so on. So,
and wasn't Emmanuelle Charpentier from Quickfire? She was at Lund when she discovered it, right?
This I don't know.
She was in Sweden, but I think she was in Lund.
Okay. You know more than me.
No, and the crazy story is that because she discovered and patented Quickfire in Sweden.
Oh, yeah, BioNTech. Yeah.
So she has a holding company that manage all the commercial rights for Quickfire, and this is
a big thing. A big thing. Yeah. I've actually I've had to work together with a BioNTech person in another board in another company. So, yes.
And we had on the show the CEO of Quickfire Therapeutics, which is the company that Emmanuelle Charpentier founded as a scientific founder, and it was I think episode 6 or something. It was pretty cool. Yeah. Okay. And
and maybe I should. We spoke about the team, Björn and Raoul, and so on. And I think at some point maybe we'll go into my background and so on. But I think there are other people, of course, in the team. I was lucky to be able to, during the downturn of the consultancy business in Sweden, I was lucky to be able to hire, and I've hired 4 people out of, really good people out of a consultancy company here in Sweden.
Is you at PwC, right?
Well, yeah, but that's not the case. This was when I worked with Cereno. I'm very much for working with enablers, because if you have your vision, you have a core team, then you work with enablers that are really good at what they do, so you get things done. And so I worked early on with banks to get the perspectives and business plan together in a format and so on very early. But then I worked with people on strategy, competitive intelligence, communication, etc., through a company called Monocle in Gothenburg. And in the consultancy crisis, biotech crisis in 2021, they had to close down. And I had been working with them for a couple of years. So I had the first chance to hire, and I hired 4 people directly out of that, very, very good people. And they are with the company and are doing tremendously well. So they should not be forgotten. I also have Nick Oakes from Astra, preclinical, head of preclinical, and Fredrik Frick, head of clinical operations. But I think another part of the team is the scientific advisory board, which we might, come to, because I think that by itself is a strong, show of commitment to what we try to do.
It's a good transition, and also I mentioned we would go deeper into repurposing, and I think on your scientific advisor board you had someone and quite some experience in that space.
Exactly.
So that we can expand there.
Maybe we can go there. And I started as a sales rep. I'm educated here in Lund, and I was questioning, should I, convert from chemistry to medicine, become a medical doctor, because I really like to understand why you get sick and what you can do about it. And then I studied biochemistry, physiology, and so on. And there was not at that time really the in-between educations, biomedical education and so on. It was chemistry or medicine. And so, I got some good advice. You've studied now the basic science here. Why don't you go and learn business? So I contacted Astra and became a sales rep. And then I met Gunnar Olsson, a young professor who's now on Cereno's board, who educated the reps. And fast forward,
Biotech is a long-term game, right?
Yes, long-term game. You fast forward to when I was recruited from Sweden to Chicago, and Monsanto Pharma. Monsanto acquired G.G. Searle, a successful family company, global company in many areas, including cardiovascular. And I was there as a young product manager from Sweden. And after a while realized together with a colleague that I was product manager and he was a medical doctor for a compound that was discovered and launched in the 60s, an MR antagonist, spironolactone. And nobody was really talking about it. And we defined the opportunity that we could improve survival with this whole drug if it was used differently in heart failure. And we went to the American Congress of Cardiology. Everybody was talking about something else. So we decided to pursue this. I couldn't convince the head of R&D to fund it. And eventually I became head of marketing globally. And I had my own budget together with my president, international operations, who just decided to pay for it ourselves. So we pursued a trial called RALS in heart failure, global trial. And we asked Dr. Bertram Pitt, now Professor Emeritus at University of Michigan, to be the principal investigator and pursue this vision with us. And we did so globally. Curiosity, Dr. Bertram Pitt is chairman of Cereno Scientific Advisory Board today. What happened with this study was that we improved survival with more than 30%. Published New England Journal of Medicine, great success. And today, more than 90% are using that medicine to help patients to live longer and live better in heart failure. I was recruited back to AstraZeneca in 95 and got in charge of beta blocker and a couple of other drugs in the cardiovascular field. I re-met Dr. Gunnar Olsson then, Professor Gunnar Olsson, who was a big boss at Astra in globally then. And we both agreed to pursue beta blocker, metoprolol, toprolol XL or Seloken for heart failure, because there was some new information that maybe also the adrenergic drive should be tempered in heart failure and could help the patients. So that time we got the financing quicker. We did a global trial with more than 4,000 patients. And these two trials at Searle, Monsanto, and at Astra finished the same year. They both improved survival with more than 30% and quality of life, and were both publishing New England Journal of Medicine. So beta blockers, when we started, were contraindicated, were not allowed to be used in heart failure. But we had enough information to say, this is going to work. Now, you fast forward again to when I met Cereno in 2014, with a vision to imply epigenetic modulation with a drug that's been in man for 60 years, that is doing something different than it was thought to do originally, something more broad and maybe much more important, HDACi inhibition. And it was like a déjà vu for me. And, we decided to pursue the journey we are on. I, at some point, was asked by Dr. Pitt, I hadn't spoken to him in 20 years, said, what are you doing nowadays, Sten? And I said, I'm doing this. We definitely should meet. Come, when are you in US? Well, in December. Let's meet. So we met in December 2018, 13th of December, and he said, this is big. We should put a scientific advisory board together. And, there's so much information on this concept. Let's work together. So that's how we formed. Dr. Pitt became chairman. Fayez Sanad, who was in both of these trials that I mentioned before, professor from France. He's also a big name in heart failure, joined. They also said, why don't you ask Deepak Bath, who's currently in charge of Mount Sinai and 280 cardiologists reporting to him in New York, 8 hospitals, big name in cardiology, editor for Brownwald's Heart Cardiology Bible, and on the board of BMS. He joined, So, and Gordon Williams, who is also a person from the past. So they joined, and they were part of reshaping the effort into orphan diseases and the belief that we can have disease modification impacting fibrosis, inflammation, myocyte remodeling, and helping to slow and maybe halt progression in these patients, both in cardiovascular and in lung. And the list of indications is longer than PH and IPF. You have heart failure, you have a number of other indications that you could pursue. So we have just started with this, and that's also why we did the second CSO14 to be able to approach the board. So these people are on the board, a scientific advisory board, and we have, in addition, we have Don de Behedzi from argenx as an advisor to the board and to operations. So I think we have quite a strong group supporting what we try to do. And we're very happy with that. And we haven't disclosed yet who's going to be working with us on the PH trial, but I don't think anyone will be disappointed. This is, we feel that there is a gravity towards what we do. Now we just need to get it done, right?
Get it done, yeah. I want one last question before quick-fire. So, I'm seeing the time. Just a quick question, quick answer. But the last thing, maybe briefly, but on the repurposing, I'm always curious because I think over the last 10 years, first, I haven't heard about too many success stories in repurposing generally, just out of my mind, out of my head. But I've heard quite a few failures as well, and I've heard quite a few pushbacks, especially from VCs who don't like it too much. I guess pharmas don't like it too much, or maybe it's very connected to the VCs as well. So how do you why is that in general? How do you look at it?
Yeah, I think there are various ways to look at this. I think, the reflection backwards on my own history here is that, the head of R&D didn't want to do it. How do you get the gold star if you're head of R&D? New chemical entities. Yeah. You don't get it for pursuing trials on drugs that are on the market. So that's one reason.
But if you can patent them and you have a clinical outcome you have sales.
Yes. Yeah, but in those days, decisions weren't made, business decisions, even at Astra, the second decision, we can come to that, but the decisions were not made business-wise necessarily. You have an R&D budget that's pursued to get those goals done. Then you have a sales market and market operation. And those... Fast forward to today, I think pharma is much better taking business decision. They have business owners of franchises in, GI, rheumatology, pulmonary diseases, etc. So I think that's better. But even when I was at Astra, we actually had to force the decision and bring the decision up to the CEO of Astra because of the heads of R&D and the heads of sales didn't have a business person responsible for portfolio investments. So I think that's historically that's been a reason. I also think that repurposing efforts should preferably be done when you have orphan diseases to pursue, because you can cover your exclusivity with orphan disease designation on top of whatever patents you pursue. So that's what we have done with CS1. But the reason to do CSO14 is actually to open up for new chemical entity deals on the same concept. So, and a broader concept. So, you could do a deal with Cereno on PH with a drug that's orphan drug protected and patent protected. That's one deal. You can do a deal on the HDAC portfolio, or you can do a deal on CS014, which is a platform deal for multiple indications. For that, you need an NCE. So I think that that's, the strategic reason why we are pursuing a historic thought. And then in addition, you can maybe take Caliditas as a successful example that pursued all the way to market budesonide, a generic substance, into an orphan disease and was later acquired quite recently. A very good journey.
Over $1 billion.
Yes. So I think, and I've spoken to the CEO who was very instrumental in that growth and deal, René, as you mentioned before, about that journey, because we are doing a similar journey with CS1. So, yeah.
Okay, I like that. Yeah. And I guess it's not necessarily repurposing that is bad. I guess within repurposing, I mean, there's some repurposing that's probably bad.
Yes,
but exactly. Within repurposing, you have some
to discriminate between them, I think.
Yeah.
But, so you can, another example that's an internal repurposing, that was the one that I mentioned with the beta blocker at Astra. The sales at that time was $200 million or 2 billion Swedish. And they wanted to close down investment. And there was 70% margin on the sales. After this heart failure trial, sales went to plus $2 billion per year in sales. And if you calculate that on a patent life that's another 10, 12 years, which it was at the time, then that's a tremendous commercial success. So as long as you are protected, what you're doing, you can take a mode of action and move it into a different disease. If you are smart about it, you can also do a new chemical entity, which I think we have been smart about, So, yeah.
I like that. Okay, a few quick questions to wrap up. The first one, and I think you have answered a bit, but what's the most common misconceptions that investors have about Cereno? One misconception.
One misconception. I think, pursuing a paradigm shift, or like we believe we're pioneering this effort into these patients and helping them. There are a couple of things you need to achieve. You need to achieve an understanding around the growing conviction in many disease areas that epigenetics is a key player. And I think that's a major force. We don't control it. It's happening. So I think, but that is a challenge. I think.
You think investors are not convinced about epigenetics?
No, I think they are. They need to understand it. Okay. I think it's much easier for a cancer therapy that's a very targeted approach. Look here, we bind here. Here's the outcome. So I think in general, cardiovascular disease has been more difficult to predict. I've been successful twice in heart failure, which has been a graveyard for many. But by realizing, understanding the concepts, and I think Cereno is understanding the concept around epigenetics and HDACs way before most people, scientists or business people in the world. So I think one of the things that has to be done to get people to understand this is actually to understand the literature that's out there, the documentations that there already, the Nature publication that happened last year, and the growing evidence that's pursued in this direction. I think the other thing has been, when I've been with this 11 years, so when you start out, you're always viewed as a little player, the small one. So for investors in the US or big investors or so, there is a price, there's an investment size, a volume that they want to do. So if you want to do a deal, any kind of deal, it's about the same effort, a small and a large deal. Most investors want to do a large investment. They can't do that in a small cap company. So you have to grow to a certain size to get their interest, to listen and understand the potential.
Okay.
So I think the size is not really a misconception. I think more has been a size. But again, we were recognized as one of the shortlisted, one of the companies of the year in Europe awards in London last year, and I was the CEO, shortlisted for CEO of the year among 10. 2 of the companies on the company award were acquired last year after that award. So I think we are succeeding in being noticed. And we were fully booked again at JP Morgan. So we're getting there.
Sounds good.
Yeah.
Is 2026 the year the biotech winter will end?
The year
the biotech winter will end?
Yeah, it's a good question, I think sometimes science, I like science, understanding, what's happening in science or in other areas. And I think just being at JP Morgan this year, there are a few, people have written about this, of course, already, summarizing, what was going on. But I think the energy at the conference was higher than the year before and several years before. So I think what's happening is that, and there are other factors going on, such as interest rates coming down. Mm-hmm. So, for a number of years, VCs have been taking care of their own portfolio, if they had money even to that. Yes. So they haven't really been looking outside, but more inside. And the people putting funds into their funds, money into their funds, have been looking to put money in interest returning papers. So I think that's changing. So the macroeconomics are changing. I think there's a key, platform of pharma having tons of money that they need to spend and having very significant patent cliffs coming forward. And there is a movement towards fundamenta, delivery, clinical assets, small molecules. So I think that's happening, and that's playing in favor of Cereno. So I like that.
2026 will be the year.
I think it will be. I don't think there necessarily will be a dramatic increase in valuations, and so I think there will be a number of deals on clinical assets or platforms. We believe we are both. So I think that will happen. I think there will be a careful investment climate still. But I think whatever asset is recognized to have a significant potential value will be taken by either investors or by pharma.
Yeah. Last quick question. One advice to 40 years old Sten.
It's a really good question. So, I think maybe, if you want to pursue a journey like this, maybe try to get the company established in the US. The journey will be less spoon-fed.
Yeah.
And I think a concept like this, if we had established this company a couple of years ago in the US, we would have gotten $100 million or $200 million as a Series A, like is happening now, actually, for unproven ideas, but just had a great potential. So I think having established companies or working with companies such as Cereno here is a long journey, and it's a hard one. But the other advice, I think, keep your friends from the past close, keep in contact. The life, when you're young, the life is concave. Everything is outward and spreading. When you get to be a little older, life is convex. Things are gravitating back to you in various parts of life, including your kids, maybe. But so when they grow older, but such as your relationships around the world, which has been very useful for me. And I think also for Cereno in this case. So I think keep your friends, keep your contacts, and maybe start in the US. I wouldn't say there are other parts of the world, of course.
Yeah. Good one. Thanks, Sten. Thanks for the discussion. Sure.
Lena, pressure. Thanks for all the good questions and the thoughts. Appreciate it.
I'm impressed by how Sten has taken Cereno from almost inception to where it is today. I'm also impressed by his persistence and more uncommon path in pharma. If you've enjoyed this episode, please hit the like, follow, or review button. Any of these actions help a lot more people discover the podcast. If you want to see similar videos, please feel free to check our channels where we have many more. I would also be curious to hear what you think. So if you could leave a comment wherever you are or shoot me an email at Philip@Flot.bio. That's PHILIP at FLOT.io. All right. Thanks for watching to the end and see you next episode.